The lymph nodes are small, oval, or bean-shaped secondary lymphoid organs embedded in connective tissue and arrayed along lymphatic vessels. Clusters of lymph nodes are found where several lymphatic vessels converge, for example in the cervical, axillary, and inguinal regions. Click the lymph nodes in the axilla, or armpit, for a closer view. The functions of the lymph nodes are to filter the lymph by removing antigens and other debris that may have entered the lymph, and to enable B and T cells to be activated by antigens. These interactions generate immune responses. Lymphatic vessels feed into a single node on its convex side. Click an afferent lymphatic vessel. Vessels that carry lymph away from a lymph node are called efferent lymphatic vessels. There are fewer efferent than afferent lymphatic vessels per node. This slows the flow of lymph through the node, allowing for more thorough cleansing as lymph percolates through passages lined with dendritic cells, lymphocytes, and macrophages. Lymph is filtered by several nodes before it is emptied into the venous circulation. Click the efferent lymphatic vessel. Sectional view. Lymph nodes are covered by a dense connective tissue capsule. Click the capsule. Lymph nodes are separated into sections by bundles of collagen fibers called trabeculae that extend from the capsule deep into the node. Click a trabecula. Beneath the capsule is the subcapsular sinus. Click the subcapsular sinus. This is the first of a series of sinuses, interconnected dilated channels, through which the lymph flows as it passes through the lymph node. Lymph from the afferent lymphatic vessel empties into the subcapsular sinus and then flows into sinuses in the outer cortex. Click an afferent lymphatic to see the direction of lymph flow. The outer cortex of the lymph node is the area just below the subcapsular sinus. Click the outer cortex. Here, B cells are found organized into oval-shaped collections of cells called lymphoid follicles. Click a lymphoid follicle. Some of the follicles contain lighter staining central areas called germinal centers. These are formed by B cells proliferating in response to antigen. Click a germinal center. Moving inward from the outer cortex, we reach the deep cortex. Click the deep cortex. Lymphocytes exit blood vessels and enter lymph nodes in the deep cortex. Here, T cells encounter antigens presented by dendritic cells. The central area of the node is the medulla. It is shaped into elongated masses of cells called medullary cords, around which lymph flows. Click the medullary cords. Medullary cords contain both types of lymphocytes, as well as macrophages and plasma cells, which are derived from B cells and are antibody-producing factories. Click the medulla. Efferent lymphatic vessels and blood vessels are found at a shallow indentation called the hilum. Click the hilum. Click an afferent lymphatic vessel to begin the flow of lymph through the lymph node. Click an afferent lymphatic to repeat this animation. Now, let's look at the spleen. The largest of the lymphoid organs, the spleen is a fist-sized, blood-rich organ located to the left of and dorsal to the stomach. It performs the same cleansing function for the blood as the lymph nodes do for the lymph. The spleen removes pathogens and aged erythrocytes and platelets from the blood, stores platelets and breakdown products of red blood cells, and provides a site for the interaction of lymphocytes with antigens. As you explore the structure of the spleen, notice how this structure allows intimate contact between blood and lymphocytes, just as the structure of lymph nodes is designed for intimate contact between lymph and lymphocytes. The spleen is a highly vascular organ. Click the spleen to explore its structure. Multiple branches of the splenic artery and vein enter and leave the spleen at the hilum. Click the hilum to see a cross-section of the spleen. Just like the lymph nodes, the spleen is surrounded by a fibrous capsule with extensions of connective tissue called trabeculae that extend into the organ. Click the capsule. Most of the spleen is made of red pulp, which looks dark in fresh splenic tissue. Click the spleen to highlight the red pulp. Areas of red pulp are sites where filtering and processing of red blood cells and pathogens occur. Lighter colored islands of white pulp surround the central arteries. White pulp is made up of collections of lymphocytes. Click the spleen to highlight the white pulp. Since one purpose of the spleen is to clean the blood of blood-borne pathogens and antigens, let's follow a red blood cell through the spleen. Blood enters the spleen through multiple branches of the splenic artery. Following the connective tissue that divides the spleen into lobes, the splenic artery divides several times, eventually forming central arteries surrounded by white pulp. The central arteries divide further into smaller arterioles that carry blood deeper into the red pulp of the spleen. Blood leaves the arterioles and filters directly into the red pulp. The red pulp consists of a network of reticular fibers studded with fibroblasts and macrophages called the splenic cords. Blood trickles through the spaces within these cords and squeezes into the venous sinusoids, thus returning to the venous circulation. Click an artery to see the blood flow again. Click a vein to continue. The macrophages of the splenic cords and venous sinusoids recognize and remove aged erythrocytes and platelets. In addition, older erythrocytes are often too stiff and fragile to successfully squeeze back into the sinusoids. Instead, they break into fragments, which are then cleaned up by the splenic macrophages. Click the image to see a photomicrograph of the spleen. In stained splenic tissue, such as we are looking at here, the white pulp actually looks darker than the red pulp because of the many darkly staining nuclei of densely packed lymphocytes. Click the white pulp. The white pulp consists of B cells and T cells. The B cells are mostly in follicles, some of which have lighter staining germinal centers. B and T cells wander through the white pulp where they encounter their antigens. Thus, while the red pulp is primarily responsible for removing old erythrocytes, the white pulp is the site of immune interactions between antigens and lymphocytes. The mucosal surfaces of the digestive tract, as well as the respiratory tract and genitourinary systems, are vulnerable to invasion by pathogens because they are directly exposed to the external environment. Like guardhouses and watchtowers strung along a castle wall, we have collections of lymphoid tissue, called the mucosa-associated lymphoid tissues, MALT, strategically distributed throughout the mucosae. MALT includes the tonsils, appendix, and Peyer's patches of the small intestine, as well as more diffuse collections of cells in the respiratory tracts and other mucosae. These tissues are unencapsulated or partially encapsulated collections of lymphocytes. MALT contains both B and T cells, with the B cells occurring in lymphoid follicles similar to those found in lymph nodes and the spleen. We will examine the tonsils, appendix, and Peyer's patches in turn. Two palatine tonsils are found at the posterior end of the oral cavity. Click the palatine tonsil. The pharyngeal tonsil, also called adenoids, is embedded in the wall of the nasopharynx. Click the pharyngeal tonsil. The lingual tonsil is a collection of lymphoid nodules at the base of the tongue. Click the lingual tonsil. Click the pharyngeal tonsil to see its structure. The tonsils trap microorganisms that enter the body through the oral or nasal cavities. Microorganisms, carried deep into the tonsils via the crypts, stimulate an immune response and lead to the formation of abundant germinal centers, reflecting the activation of B cells. Click a tonsillar crypt to see the path that microorganisms take. Sometimes tonsils can be overwhelmed by bacteria and become sites that harbor rather than kill the organisms. The vermiform appendix is a blind sac stemming from the wall of the first part of the large intestine. Its walls contain a large concentration of lymphoid follicles. Like the tonsils, the appendix can be overwhelmed by bacteria. In some cases, its mouth can become plugged by debris. When this happens, pressure within the appendix grows as pathogens multiply within it. This is called appendicitis. Click the appendix to give this person appendicitis. A ruptured appendix can be life-threatening because it spills pathogens into the otherwise sterile peritoneal cavity. Surgical removal of the appendix and its spilled contents is required immediately. High doses of antibiotics are used to kill any remaining spilled bacteria. Peyer's patches are found in the mucosa of the distal portion of the small intestine. Click a Peyer's patch. In this photomicrograph, you can see the many lymphoid follicles in the small intestine that make up the Peyer's patches. Like the tonsils and other MALT components, Peyer's patches are located where they can sample the antigens moving through hollow organs open to the external environment. If a pathogen escapes the defenses of the MALT, it can still be cleared by responses in lymph nodes or the spleen. We have now finished our investigation of MALT, the last of the secondary lymphoid tissues and organs that we will consider. Now let's look at the thymus, a primary lymphoid organ. The thymus is the site for differentiation of lymphocytes into mature T cells. Thymic hormones and other factors influence the development of immature T cells. Click the thymus to begin. The thymus is a bilobed organ located in the mediastinum. In young children, the thymus is large relative to body size. The relative size of the thymus, as well as its function, gradually decreases with age. In the elderly, thymic epithelial cells are almost entirely replaced by fat cells and fibrous connective tissue. This process, called thymic atrophy, may be one reason why the elderly are more susceptible to infection. Click the thymus to see thymic atrophy. Click the thymus to view the organ more closely. Each lobe of the thymus is divided into many lobules. Each lobule contains an outer cortex and an inner medulla. Most cells in the thymus are immature T cells at various stages of development. Scattered amongst the immature T cells are thymic epithelial cells, which influence T cell development and secrete